Hi.
Hello.
Spence, would you like me to share the screen or are you going to share the screen?
If you can share it, that would be great.
Okay.
I guess you'll just have to advance it, right?
Yeah.
Okay.
Let's wait a little bit more time. I think we are pretty much, that's the people that who, okay. And first of all, thank you very much for everybody to put some comment on the Lisa's card. It was so nice to see everyone's short kind of greeting for her. I think definitely she feel it very glad to get that because she emailed to me and also the Barani saying that it was surprised to see that kind of card coming from all trainees from different institutions. So thank you very much, guys. Thank you very much for that, doing for it.
Thank you for organizing, Tanya.
Happy to do that. So today, I think we can start. First of all, today we are a little bit different and we will have a short, very short presentation from Dr. Bemmer introducing our Labswap program. This is the structure that we are planning to implement. It is still in the process. So we are happy to have any feedback if you have it. But we are giving this information in advance for you because I think it's need a little bit planning between your primary mentors and also your schedule, because we know that course started differently. Each institute have different requirements. Therefore, we sharing information in advance. You have plenty of time to think about. Dr. Bama will explain it. But just to give an overview, so this is not final. So you can share any feedback if you have. So I will go ahead and ask, we will start. So let me share my screen. Everyone can see my screen. Okay, so Okay, Spence, you can go ahead.
Okay, thanks, Tanya. Thanks everybody for being here. I'm gonna just kind of, we put together, Tanya and I have worked on this over the last few weeks. And this is a simple overview of the program. And we're gonna start with the objectives of the Labswap program. The primary objective is for each of you to have a collaborative experience with another BPRI institutional member, possibly even a trainee, if you decide to do a collaborative project. And the focus for this is really to expand your current research objectives related to your PhD or your master's. This is not about creating something completely new. It's about working with another BPRI member to expand sort of your current area of research beyond what you're able to do in the lab that you are pursuing your PhD. Two other aspects that are critical to this is that we want you to experience a different working environment. And the idea here is for you to just to see how other labs are run while you do this exchange program. And then another really critical part, and this is important for your development and for future prospects, it's an opportunity for you to expand your scientific networks. And so what do we mean by this? When you go to somebody else's lab, not only do you get to interact with those lab members, but you will be interacting with potentially lab members in the lab next door, lab members in that department. With this Labswap, you will physically be in another location, and this is going to give you an opportunity to interact with other members of that department that you're visiting. What kind of opportunities might present themselves? Other departments will have seminar series, and so you'll have opportunities to engage in those seminars. There will be events in the department where you'll get to meet people. And the other critical thing that we would really like you to focus on as you're thinking about what group would be most appropriate for you to do the Labswap with is, are there people in the department of that BPRI member that might be valuable in terms of exposure to ideas? Maybe there are things that you're interested in and there's a researcher in that department that you want to do the Labswap in that have these expertise and skills and experience. And so when you're looking at these other labs, sort of think broadly about which BPRI lab is going to afford you some really great opportunities to sort of build on your research program. And sort of think about it in these three levels. Okay, next slide. What's the overview of the proposed program? We've given this a tremendous amount of thought. And again, we want to try to keep it simple, not to overburden you with lots of requirements. But the idea is for each of you to develop a very short proposal, and I'll go through the sort of the details of this shortly. And you'll develop this proposal with your PI as well as the PI of the Labswap that you will be working in. We're going to lay out exactly sort of how this process will work. And a key way to think about this as we were putting this together is how does anybody do collaborative research? Collaborative research often comes about because you've seen somebody give a talk and you thought, oh, that was really interesting. I have research ideas that kind of align with that. I'm going to go talk to that person, sort of pick their brain for ideas. Maybe there's an opportunity for us to sort of combine our research. And the only way you can sort of begin that process is to have a first discussion and find out what the possibilities are. So we're going to try to encourage you to sort of think about treating this like any collaborative experience. Somebody has something in their lab that they're doing, you're interested. How do you initiate a discussion? And then how do you sort of go towards actually developing some collaborative project together? Once this proposal's been written, it will go to an ad hoc committee of BPRI members to be reviewed. The idea is not to be overly critical about this, but just basically to make sure that things are aligned and on track. And effectively, if you've worked on this proposal together with your PI and the PI in the other lab, you should be in great shape. We see this year, or this project lasting for a year and sort of existing minimum of a year. It may go longer, but it's going to exist of 3 stages. The pre-planning stage, which is putting together this proposal and thinking about what the project might look like. And then there's actually going to be the conducting of this collaborative work. And sort of here we're thinking maximum 2.5 months. And then, of course, after you've completed the actual work, you'll have data analysis, writing up to do, putting together a manuscript. And so that's sort of the post-training. So again, data analysis and manuscript writing. And again, there's a couple ways you can do this. You can just work to extend your own project, but maybe you see another trainee that's doing something, and maybe you can envision working together with that other trainee to develop sort of a deeper collaborative project with another BPRI trainee. So if you want to sort of build with somebody else, you can do that. But if you want to just build yourself on your own research, that's fine too. Does anybody have any questions? If not, there'll be plenty of times for questions at the end. So next slide. This is sort of the timeline and sort of the mechanisms we see the program running. And a total of 8 steps. The first step is for each of you to identify a potential Labswap mentor. And I think a number of you are clearly already sort of down that path. The second step is, once that person has been identified, it will be important for you plus your PI to have a meeting with that Labswap mentor to have your initial discussion about possibilities of projects. Once you've had that meeting with your PI plus the Labswap PI, you'll go about drafting the outline of a research plan. Once that is drafted, you'll meet with your primary mentor and your Labswap to discuss the outline, right? And so you're going to be getting feedback on, yeah, this looks good, but maybe this second thing, we might need to think about it slightly differently. But it's to get some feedback. And then you'll write and submit the full proposal once you've had feedback from your PI and your mentor PI. It will go to the research committee for review. Once it's approved, the interdisciplinary training will begin. And then the last step will be the production of your analyzing your data and writing it up. Okay, and again, sort of think about this not as you have to have a completely new novel standalone project, but this is sort of an add-on how you're sort of extending the research that you're currently conducting, but working with somebody to add a new novel component to what you're doing. Okay, you're not reinventing the wheel, you're not creating a wheel, you're not starting over from scratch. Think of this as extending the work and the ideas that you're currently working on. Okay, next slide. Proposal and review criteria. So this will be important to you guys. And again, we have another document that provides the full details of what this proposal structure looks like. We're not quite ready to give it to you yet, but we wanted to share with you sort of our vision of what this proposal will look like. Again, it's not going to be a massive amount of work. That's not the focus here. The focus is to develop ideas and keep things simple. So the proposal structure will have a brief summary about what the project's going to be. You'll then have a longer project description, and we put word limits on this, so they won't be massive. They're not going to be sort of 5-6 page things. They're sort of 1 page, 1.5 page project descriptions. The project description will provide the objectives, explanation of your visit. It will talk about your collaboration or integration plan, team role. and then what are the expected outcomes. There'll be a small section that will provide an explanation of how this Labswap will benefit your career. There'll be a timeline component, again, sort of working together with you and your advisor and your Labswap mentor. What's the timeline that works for everybody? We're providing money to support these projects, including travel costs and accommodation costs, those sorts of things. And then we'll need a one-page bio sketch that includes lists of publications if you have any yet. So that's the proposal structure. Again, it may look long, but it's actually not, and it's all pretty straightforward. And I think it'll be relatively an easy task for each of you to complete. In terms of the review criteria, the ad hoc committee just wants to see a clear explanation of the uniqueness and innovations of the proposed activities. And again, you're going to be working within the 10 research themes of the BPRI. They will evaluate the plan for the collaboration and integration, effective funds and utilization to improve the outcomes, a clear plan for publications. And again, why we keep hammering on the publications, it's important for two reasons. The first and most important one is important for you because in order for you to demonstrate that you are sort of on this sort of path and putting yourself in a position to be attractive to labs or industry or whatever your individual career goals are, the best way to sort of demonstrate your effectiveness and competence in terms of generating data is to have a publication. The second reason this is important, we need to sort of have outputs that we can report to NSF, and the best and strongest output that we can report in every case is a publication. And then the last thing that they will sort of look at is contribution for trainee career development. Okay, next slide. So what should you do? And I think this is our last slide. Is this right, Tanya? Here's what we ask you to do now. And again, I think many of you have done this already. Review the faculty members' research interests. The links are available on the BPRI website. So go forward, look through the entire list of faculty and PIs and decide which lab would be a good lab for you to collaborate with to expand your current research. Think about an interesting research area that you can integrate, and that's in most cases, you know, is a logical, natural extension. Decide the lab that you want to visit, and then identify an action plan for creating a proposal for that interdisciplinary training. As you're sort of going through the website and looking for potential Labswap mentors, once you've had this opportunity and you've identified one or two, I would strongly encourage each of you to have a conversation with your main PI, just to sort of let them know your intentions and your thoughts and the directions that you're going, get their feedback. And then once you're both in agreement, or your PI is in agreement and says, yeah, that would be a good Labswap mentor, then the next step will be to reach out to that Labswap mentor. I think that's it, right, Tanya? Is there anything I missed?
I need to add one thing just to clarify why we go with the grant proposal format. Just because if you have in your CV, you got the grant from somewhere, it looked nicer for you. Yeah. Even though this is internal, not have competition, real competition, we want you to show to other people you got a grant from somewhere else. So this make your CV look nicer. That's why we come up with this idea rather than asking, okay, let's go to the other lab and do that. And also having plan allow you to have your time effectively used in the duration you visit, and also the Labswap mentor have clear idea what you are going to do in his or her lab. And also remember that when you go into another lab, there are some official protocol to follow. So having such documents help them to allow get your to that university and spend 1 or 2 months in there. So that's why we have that, not to add you another task, but make you to this task benefit to you at the end. That is why we are planning. So if you have any feedback, if you like to receive any feedback, or if you like a little bit different suggestion, please feel free to share with us. We are open to your suggestion too. We are not saying that what we propose have to do in such way, but if you have any suggestion, please share with us.
I guess we can stop sharing the screen.
Yes.
Anybody have any questions or comments at this point? Again, I know I can speak at least for Chris and Arani. We've talked about this already, and we've kind of identified a potential lab. So I think some of you, have been already thinking about this. I don't see Alexis on, but she's actually probably going to be coming to Texas A&M this summer. Is she on? Oh, sorry, there you are. Hi, Alexis. And again, we'll have the sort of full details of the what needs to be in the proposal, including the sort of different sections. And again, I just want to emphasize we've tried to keep it simple and short as possible. So hopefully this won't require a tremendous amount of effort on your parts. And Tanya is absolutely right. This is another bit of information, bit of data that can go on your CVs, and demonstrating that you're successful in getting funding, whether it's internal or external, is always a good thing to have on your CV.
Can I ask a question regarding the, to help the student identify the research interest? So on the website, there is indeed research interest of PI, but do you have like a document like the same way you created for the trainees, Tanya, for the PI, that is a bit more complete than what you have on the website?
Not yet, but we are working on a kind of creating video series and showing what each lab doing, especially before the boot camp. We will have some more solid information about each lab, what they are going to do. At least we are trying to have like 3 to 5 minutes short presentation explaining each lab, what is they are going to do in BPRI and what is the interesting area that student can see in their labs. So basically Tanya, by boot camp, you will have a good idea.
Tanya, do you remember what our expected sort of timeline on this is?
So the expected timeline is why we're giving at this stage for you to is you need to plan ahead, especially for the PhD student. We don't know where your coursework, everything is finished. So we just need which year you like to go to the lab visit and which lab by end of October. So in that case, we have a clear idea for the budget arrangement and everything for the next year, and we can plan accordingly. That's why we are asking by October, because BPRI fund cycles end at each October in each year. So we can, if you get whole detail to us by October, we can plan according to next year. Okay, we have 2 trainees go for lab visit, so they need funding, and this will be how we given the funding for them.
Really quickly, I'm guessing, so yeah, October is sort of just the date when you need to let us know when you want to go, and then it'll be the process of then working on the proposal. I'm guessing pretty much everybody here is going to be attending the boot camp. One of the things that we're working on for that itinerary is to have plenty of opportunities for sort of social interaction. And the very first day, the plan is to spend time together touring the facilities. And, you guys, know each other, reasonably well through Zoom, but it's obviously much better to be in person. And so our hope with the boot camp is that first morning, is to really sort of have an opportunity for people to hang out and interact together rather than sitting at desks and having presentations. The idea is to sort of visit the facilities, have some interactions, so that we really get a chance to engage with each other in a more meaningful way. And then hopefully that too will sort of grease the wheels for the rest of the boot camp when we do have to sit down and be forced to kind of sit and listen to presentations.
And one thing I need to correct it for the Alexis, you have different timelines. So don't worry about October, everything. So you have whatever your timeline, we are happy to support you. So your case is different. So just let you know that Alexis is master's student. So we are offering her training in advance to other people who are PhD students mostly.
Yeah, and so we've kind of joked about this with Alexis a little bit. She's kind of the guinea pig for this opportunity, and so we'll sort of learn through her a little bit, sort of if there's anything that we need to do differently. But we're excited about hosting her this summer. Maya is going to be involved in this project as well, and I think it's going to be a lot of fun.
Okay.
Okay. If anybody has any questions, feel free to reach out to Tanya. Feel free to reach out to me. Again, we will have the details of the program available probably before the boot camp, I would think.
Yes.
But the big issue, right, is that there won't be any urgency for you guys to sort of write these proposals. The key thing is making a decision by October about when you plan to do your Labswap. So lots of time to think about this. No rush. Okay.
Sure. Thank you, Spence. I know that you have to leave. So shall we go with the research presentation?
Okay. Bye, everybody.
Bye. Arani, do you like to go first? I thought you may be late, or do you like to go a little bit later? It's up to you. Let me know.
I can go now. That's fine.
Okay.
Yeah. Let me just pull up my PowerPoint. Okay. Do you guys have the presenter view, or does it look okay?
Presenter view.
Presenter view.
There we go. That's better, right?
Yeah.
Okay. So for my project, I'm going to be just talking about like the first half of it, or what I'm currently working on. So phase 1 of my research project. Right now, I'm looking at thermal tolerance across all of the different species of Schistocerca that we have here at Texas A&M. So my question is, does thermal tolerance of Schistocerca differ across phases? So solitarius gregarious, life stage, second instar, final, and adult, and then species and sex, so male and females. So I only listed 2 hypotheses here. My first one being that solitarius individuals will have a higher LT50. So LT50 refers to lethal temperature of 50% of the population. And then, so higher LT50 than their gregarious counterparts. And then my second hypothesis is that second instar nymphs will be the most susceptible to temperature changes resulting in lower lethal temperature or LT50s than the other 2 life stages that I'm testing. So in order to do this, I plan to, my slides won't move, there we go. Randomly select 18 females and 18 males for a total of 36 per temperature that I will be testing. I'm going to place them in a falcon tube with a moist cotton ball to control for desiccation and allow them to acclimate for a total of 15 minutes. Then I'm going to place them in a recirculating water bath at one of the pre-designated test temperatures for 1 hour. So this is an acute stress. I'm testing temperatures of 45 for 1 hour, 47 for 1 hour, 49, 51, and 53. 53 being what I expect the 100% mortality to occur, and 45 degrees Celsius is where I would expect 100% survivability to occur. For the different species, I'm ranging between 47 and 51 as where LT50 will occur. So where 50% of the populations will die. After the heat shock exposure, I'm going to move 15 females and 15 males to separate containers, where they will be individually monitored for survivability for 1 week. And then 3 of the females and 3 of the males will have hemolymph extracted for metabolomics, and then flash frozen for later heat shock protein analysis. So my expected results from this are, 1, to use a GLM, so generalized linear model, to fit my LT50 curve. So this is just what I modified from a different paper that I published during my master's. So proportion surviving, and then test temperatures, and then just identifying where 50% of mortality would occur. So this is from like I said, a different experiment, but for example, 50% would be where this 0.5 is, and then you would just follow the line and say that, for this particular species, which in this case would be Cuevas-Sanchez, yeah, the LT50 would occur at about 47.3 degrees Celsius probably. From that, I'm going to look at the heat shock protein expressions. Like I said, I'm flash freezing a total of 6 individuals after acute stress. And depending on what my results are from the LT50, whatever seems the most interesting is what I'm going to use to actually test for heat shock protein analysis. So in my hypotheses, I stated that I think solitarious individuals will have a higher thermal tolerance than gregarious ones. So if my hypothesis holds true, then I'm going to go back and pull the 6 individuals from that specific life stage and compare their heat shock protein expression in the solitarious phase to their gregarious counterparts. And this is just an image that depicts what heat shock proteins do. So under stressful conditions, usually proteins would be denatured, but when these types of stressors are detected, heat shock proteins are produced, and then they can refold denatured proteins, so they can help an organism tolerate changes in their environment. And I'll be using qPCR to do that. And then the final phase of that project is going to be to conduct field work to compare my results from laboratory reared colonies to their wild counterparts. I obviously won't be able to do that with all 6 of the species that we have here. Most likely it's going to be Americana, because we can get Americana from Florida, which are the ones we currently have in the lab, are from a line in Florida, and then Piscifrons in the Yucatan Peninsula. So, and that's what I have. And I'm happy to take any questions.
Thank you.
Yeah, I have a question about your hypothesis that the isolated would have a greater tolerance. Why do you, I'm just wondering how that developed, or how you got there.
So my thinking behind that is that heat shock proteins are expressed under stressful conditions, and being gregarious or in high densities is a stressful condition in itself. So gregarious and colonies of gregarious individuals should already be expressing a higher level of heat shock proteins. So they're not going to have as much flexibility to express additional heat shock proteins under heat stress, where individuals that have been reared solitarious haven't experienced stressful conditions. So they're going to have a wider range of heat shock protein expression available under unfavorable conditions.
I actually had a question, Arani. So when you will do the field work, and you're extracting the hemolymph, you're going to be looking for expression for the heat shock proteins. So in fieldwork, do you think that your hemolymph will remain stable for that long for you to conduct transcriptome analysis when you're back? And or do you already have that procedure done, like how you will be making sure that that it remains stable?
Yeah, so the hemolymph is going to be extracted for metabolomics. So Spence has a colleague at a different university who's going to be able to they're trying out some new methods, and so they were interested in collecting some of that hemolymph for themselves. So at the moment, that may or may not be something I may try to do in the field. What I think I'm going to target for directly in the field is just looking at using the same methodologies on just wild caught individuals and testing their thermal tolerance, and compare what I find in the field to what I found in the laboratory, and then see how realistic my results are to a natural environment.
Hi, Arani. I have a question.
Yes.
I'm not sure if you mentioned this, but do you use your heat stress assay purely as a proxy for general like stress to induce like general stressors, or are the temperatures you're using physiologically relevant to like what a locus might encounter? Just because even your lowest temperature is very, very hot, and I don't like, I wouldn't think that like even in gregarious swarms they would get that hot. Or maybe it's like, you know how bees, when they congregate in their hives, they manage to like generate a lot of heat. Like, I'm not sure how that is relevant to your study.
Yeah, so the temperatures that I chose, the gregarious individuals can experience temperatures as high as 47 degrees Celsius. And what I have been able to find in the literature is that they purposely will move out of temperatures once they reach generally above like 45 degrees Celsius. And then they expect that the lethal temperature, or LT50 for gregaria, is going to be at 51 degrees Celsius. So that's kind of how I went off of those temperatures, was that these are temperatures experienced in their natural environments, but they're definitely on the hotter end. So what may end up happening is that I might have an LT50 that is actually lower than 45 for some of the species or some of the life stages. And I may end up having to shift my test temperatures a little bit further to the left into those cooler temps.
I have a question also, although like I chat with you already often, I just thought about like, you know, the question regarding the stress. Do you think it's possible that you also measure maybe cortisol level at just like first as a baseline between your isolated and your crowded, and just see if the cortisol level of your individual, which is normally used for like stress, you know, indication in insect or in other organism, and then you can already see if your baseline is like different between an individual solitary and a crowded one. I don't know if you had envisioned that already.
So I did not. That's not, yeah, cortisol levels is not something that I had thought about. From just my training, my thoughts for stress are more behavioral based and like, you know, survivability based. But yeah, cortisone is a good way to go about that. It's not something I had considered. I wouldn't actually know how to go about collecting anything related to that.
Well, maybe with your hemolymph already, I think. But we can chat about that if you want. I just had the idea starting there.
Yeah, good point.
Okay, thank you, Arani. So can I move forward with the blessings? Blessing, are you ready? Blessing. Maybe she's not hearing us. Okay, so can I ask Margaret to go then?
Yeah, let me share my... Okay, oh, that's not the right thing. Okay. Here is the present. Anybody see the present on here? I don't know.
On the bottom of
on the bottom. Okay. Yeah, there we go. All right. Okay, so I'm gonna kind of give an overview of like previous experiments that I've done in my lab, and then how it's relevant to like the BPRI work, because I've just actually got back from Texas A&M last week where I did the first set of experiments. So I haven't gotten much done yet, but I'll kind of give you an idea of what I do here and how it's going to relate to our work. But basically our lab works on visually guided escapes. And so specifically, like I work with grasshoppers and flies, and I'll be working with mice as well, but specifically for the BPRI, this is going to be focused on the grasshoppers. So essentially what we do is we use looming stimulus to simulate escape or simulate predators approaching the grasshoppers, and we use a ratio of the half size of the simulated object to the velocity to adjust for different approach speeds. So for example, a larger L over V, which is the size over velocity, would mean a slower approach. So typically we use, for initial experiments, we would use a range between 20 and 80. So for the experiments I did last week, I used 20, 50, and 80 to give kind of like a slow, medium, and fast approach, give us a good range of those. So kind of the neural circuitry of involved in escape with grasshoppers. So here you can you see my mouse? Okay, so here we see the eye, and this is the brain. So the LGMD synapses between the visual cortex and the brain, and then there's a 1-to-1 ratio between the LGMD and the DCMD. And this DCMD goes all the way from the brain to the leg. And this is where we are recording from when we do our neural recordings. So basically, what that looks like in our lab is, this is a dissection that I haven't done in a while, but I do frequently, is right here. These are the DCMD neuron, or these are the ganglia, and the DCMD neurons are within these bundles. They're kind of, I don't know if I couldn't find a good picture, but basically each neuron, the DCMD is on the inner part, right near the surface. So you can do an extracellular recording from there, which is what this shows. These hooks are electrodes, and they are de-insulated just right in the bend to record an extracellular recording from the DCMD there. So we know that the DCMD firing profile is really well characterized, specifically with black liming stimuli on a white background. So these are some examples from another project I worked on with those same 20, 50, and 80 L over Vs. And we basically see a rapid spike right before collision, simulated collision, and then a drop that occurs right before the actual collision. So the spike we see is about approximately 100 milliseconds before the simulated collision. And in another project that somebody did in the lab, they used simultaneous chronic recording and behavior to determine like where the spike occurs versus where the behavior occurs. And we know that the behavior occurs right around when the drop is. And this instantaneous firing rate is basically how much the neuron is firing at that moment. So like I said, these are just examples, but you can kind of see, so 100 milliseconds would be here on this one. It'd be about here on this one. This one is a little bit off, but they're just examples. So the population average would be about 100 milliseconds. And then, so behavioral experiments, which is what I just got done doing at A&M last week, we use the same stimulus, and we use free behaving grasshoppers. And you can see right here, they kind of walk out onto this platform. We trigger the stimulus, and then we determine the jump timing by looking at the videos that we take. And essentially, as soon as this leg starts to expand, like when the knee starts to straighten, that is when we would record the jump time. So this is kind of a different experiment where we were comparing black versus white. And we know that with white stimuli, the responses aren't a little bit, the probability jumps are a little bit lower. And we also see a larger spread in the jump times with both slower stimulus of both colors, but specifically with white, it was significantly larger. But that's, with our BPRI research, we're mostly going to be focusing on just using black stimulus. So this isn't super relevant. I was just trying to find a good example for you guys. So basically how this all relates to the BPRI project is we are going to be, like I said, previous studies have shown that different approach speeds and accelerating versus decelerating, different types of color, black versus white, all show different responses in behavioral and electrophysiology. So the studies we want to do, and specifically with Schistocerca gregaria, but ideally with all of the species, would be to compare the escape responses and the electrophysiology recordings to these stimulus in isolated versus crowded conditions. So we hope to do eventually all of the species, but we're starting with Gregaria, and then we're going to do Americana to compare to our Americana here, make sure that, you know, control for those kinds of rearing conditions. And then if we see significant differences, then the next steps would involve kind of single cell transcriptomics to understand what the differences are that are occurring at a cellular level in those neurons. And I actually have the initial data from Texas A&M last week. So these are Gregaria on the left here. This is the L over V 20, 50, and 80 of the crowded individuals. And we're just looking at jump probability here. And then for the isolated individuals down here. So only out of, so each of these had about 60 trials, so 60 in each of these 6 groups. And out of all of these individual trials with isolated, only one single jump occurred. Meanwhile, we saw pretty normal as compared to what we see with our Americana behavior for the jump probabilities in crowded. So that's already something that's really interesting. I'm doing some data analysis right now to see how significant, but I mean, it is pretty significant, the fact that we only had one jump in all of the isolated trials. But yeah, so we're hoping to continue to do this with all the species and see kind of what else we can get out of them and see if we can maybe do different types of approaches, stuff like that. But yeah, that's pretty much where I'm at right now.
Thank you, Margaret. So anyone have comments or would like to share something, feedback? Okay, because we have some limited time, can I ask Blessing to go forward and present her presentation?
Oh, did I stop sharing? I don't know. Oh, stop sharing. Okay, there we go.
If you have questions, please type the questions so then we can answer the questions in next meeting too. Or if we have time at the end of presentation, Blessing, okay, we can see your presentation. And okay. Before Blessing starting, I would like to introduce a little bit about Blessing, because Blessing is an undergraduate student from the Southern Illinois University, not a graduate student. So when you're commenting or giving feedback, please have this in your mind.
Okay, so this is a post I presented a couple of weeks ago at LSAMP. So I'm just going to talk about like my project and what we are currently working on. So basically my lab works on characterizing the microbiome of Schistosarca cancellata. And as we've all like talked about today, like they're in 2 different phases: the solitary phase, which is like placated, harmless, and we also have the gregarious phase that like swarms and causes millions of dollars in damages. So, and most local studies have been done in labs because it's easier to study them, it's easier to like control the species. And in one particular colony of locusts that we received from our collaborators, like we noticed a pathogenic infection. So the locusts in this, like from these colonies would like undergo this like melting, and they start to twitch, and after a couple of hours they die. So basically we wanted to see like one, identify what pathogen is causing this disease, which we termed pink melting disease. And then 2, like how the pathogen is occurring in the local species. So that's just one component of my project. The second component is more about like sequencing, which is particularly for BPRI, and I'll talk about that later on. So we did PCR using like several red pathogenic bacteria, including Pseudomonas aeruginosa, Serratia marcescens, as well as some control like Enterobacter and Photorhabdus luminescens. And then using a PCR, we found that Pseudomonas aeruginosa was the pathogen infecting this lab rat colony. And once we had that discovery, PCR in a sense is limited in how much it can tell us about how the pathogen is infecting the local species. So we also wanted to do qPCR to try to quantify how much of the pathogen is present in various local species. And if there was like a certain level of pathogenic infection where the locust eventually becomes sick and dies. So we had two different hypotheses. The first one is a titer hypothesis. And what we proposed for that was that all locust samples had varying amounts of Pseudomonas aeruginosa. And then at a certain level, one of them, like maybe like a really high number, one dies and infects the rest of them. But the carrier hypothesis just says that like only a couple of locusts have the Pseudomonas aeruginosa pathogen. And then when that one becomes sick, it spreads it to the rest of them. So to test the hypothesis, we did qPCR. And this was just like a standard qPCR procedure. We had 10 microliters of cyber-safe, 7 microliters of nucleus-free water. We had the Pseudomonas aeruginosa gyrase primers. And so for the lab rat colony, we had 10 biological replicates and 3 technical replicates for each individual. And we had the same thing for our field caught individuals. So when we did the qPCR, you can see our results here, still preliminary because we're in the process of analyzing this data. So we had the CT values and we felt like we weren't able to get a very direct and like concrete interpretation from just analyzing the CT values. So we wanted a way that we can convert CT values to Pseudomonas aeruginosa cells to give us something more quantitative. So we had pure isolates of Pseudomonas aeruginosa, which we performed the serial dilution on. We also measured the DNA concentration and how many cells per milliliters was in each of them. And through doing that, we were able to get this formula that allowed us to convert like CT values to Pseudomonas aeruginosa cells. So if you look at this graph here, the lab rat colony is the one in gray, the field caught group is in this orange-ish color, and the symptomatic one, which are the ones that were actually diseased and eventually died, are the ones in this yellowish group. So something interesting that we just noticed from the beginning was that the lab rat colony had a wider, I guess, spread of Pseudomonas aeruginosa cells. There was even this one outlier that had Pseudomonas aeruginosa cells higher than what you would find in a diseased organism. And that was interesting because like it negates the, of course, it negates the carrier hypothesis because like all samples had Pseudomonas aeruginosa. But interestingly, the titer hypothesis wasn't also holding true because we had lab rat colonies with Pseudomonas aeruginosa counts really high enough, but they were also not sick. So in our proposed like explanation for that would be that there might be other factors like, you know, overcrowding, humidity, rearing conditions, and things like that that may contribute to triggering PMD. Since this doesn't seem like, you know, a linear correlation of like Pseudomonas aeruginosa present in the local species leading to triggering PMD. So that's kind of where we are in terms of that conclusion. We haven't done any further tests to like see if what kind of stressors are in play here. We just have stopped at doing the qPCR. But I think something that's really interesting with this data is that most pathogens infecting like pest species are very useful for the creation of biopesticides. So we think this would be like Pseudomonas aeruginosa would be an interesting consideration for making biopesticide in the future because it's a naturally occurring pathogen in a locust, especially for Citrosecca cancellata. So that's one ecological benefit. And after seeing like this vast difference between like the lab rat colony and the field caught colony, we thought it would be interesting to go a step further and try to see what more specific differences are there in the microbiome. Because if we're taking locals for the environments and bringing them into the lab to study them, we want to know if there are any differences in their gut microbiome that might be predictive of their behavior, or how that would affect any like experimental conclusions that we make. So we have prepped various samples. We have a lab red colony and wild caught colony from Bolivia that we have prepped, and we just did dissections, which is actually from a protocol that we developed in our lab and worked on. And after doing dissections, we had DNA extraction. And I would say one thing we do with our DNA extraction is that we try to treat our samples because they've been preserved in ethanol. So we treat them with S1 lysis buffer and proteinase K 24 hours before doing the actual DNA extraction. And that allows us to get like really clean bands and, you know, more clean DNA, as you can see from our preliminary results here. And right now we have 15 samples each for both the lab red and the wild caught colony that we've sent up to manufacturers to help with sequencing the 16 SV region. And so once we get that results, we're trying to see if there are any differences in their gut microbial composition, and basically just use that as like a base level for future experiments and how that impacts the kind of interpretation that we can make from local studies. Yeah, that's my presentation.
Thank you very much, Blessing. So anyone have comments or feedback?
Hi, Blessing. I think this, oh, sorry. Can you hear me? Oh, okay.
Yes.
This is like pretty cool work. I was just wondering, because I know that in locusts, their poop, there are a lot of pheromones in there. And so is it known to what extent the microbiome is responsible for a lot of the behavioral pheromone responses in locusts?
That's a really interesting question. So we haven't looked at pheromones really. I personally have been working on more like microbiome specific things, and not necessarily like we just always like extract the whole guts. So if there's any like specific region of their guts like impacting the microbiome, I would say we don't know that because we haven't tried to study that yet.
Sorry, what I meant was like not like, I meant the more broad definition of pheromones as in any sort of chemical, like for example, a byproduct of the microbiome metabolism, for example, and if those had any sort of behavioral significance in terms of like pheromone and like communication and things like that.
I think my answer would still be that that's not something that we've studied as far as I know. So most of our samples we get from Arizona State. So we're not able to like tell directly since most of them are preserved. And like right now we're still at the very beginning. Like this is the first like large scale genomic analysis that I'll be doing to compare the lab red and the wild caught individuals. There are some people who have worked on like different stages. So there's someone working on eggs, or like I work on the 5th instar and 6th instar nymphs. So that's as far as we've gone in terms of like analyzing behavioral differences in the S. cancellata.
Okay, thank you. So because of the time, I have to move forward with the Muchun, our BCM presenter. Can you please go ahead and present your presentation?
Yeah, sure.
He's a PhD student from the Chuck's lab.
Hello, this is Muchun from the Chuck's lab at Baylor College of Medicine. And the main focus of our lab in the BPRI is on the transcriptome side, and my project is to construct a density resolved single cell atlas of the locust brain. And in terms of the locust phase polyphenism, we will focus on the transcriptome side. And the first question we want to answer is what cell types and are present in the locus genome, in the locus spring, and we will identify the specific genes for different cell types so that the promoters can be used in further genome editing tools for mechanistic studies. And in addition to that, we will also compare the single cell transcriptome between the locus at different phases, the solitary phase and the swarming phase, so that the differentially expressed genes between the two phases would be identified so that we will know different cell types might have different responses in the phase changes. And specifically, we will use the high throughput single nucleus RNA-seq assay our lab currently developed, which we denote as METQ-JOB. METQ-JOB is compatible with fixed nuclei, and we can use the dissected tissues and directly freeze them. After that, we can homogenize the tissue to get the nuclei and fix them. Next, we will encapsulate the single nuclei with the barcode beads containing the primers into individual droplets. Within each individual droplets, there is a single bead containing primers as well as a single nuclei. So we have thousands of droplets for the multiplex reaction. Within the reaction, we will perform the barcoding of the RNAs so that next we will break the droplets and perform PCR amplification of the RNA-seq library and proceed with sequencing. Compared with the commercial 10x platforms, we not only we have three major advantages. The first is that the compatibility with the fixed nuclei. So we can use the frozen samples, and this would reduce the potential batch effect. And another is that we have significantly improved detection sensitivity compared to 10x. And more importantly, our platform is much cheaper compared to the 10x, so that in budget-wise, it can fit into the large scale study. And in the first stage, we would sequence the brain of the Schistocercus americana to generate the comprehensive cell atlas of the Schistocercus americana brain and identify the different cell types using the unsupervised clustering, and therefore identify the cell type specific genes. In the second stage, we would move forward with the Gregoria species, and from the we will sequence the brain at low and high density. And we will in the unsupervised study, we will try to identify whether there are the density unique cell clusters that would represent the phase switch. And also we will identify the cell type specific differentially expressed genes in the density changes. And this would provide from the transcriptomic side, would generate new insights into the molecular mechanism of the density switch in locus. And that's basically all about my presentation. And do you have any questions and comments?
Thank you, Muchun. So anyone have comments and any feedback for Muchun? I know that we are a little bit running off the time, so please make sure to rate today's meeting, because I'm kind of tracking it for each meeting, so how it went. So please use the link that I shared from the chat and mark your preference. There's only one question that you need to click. So many thanks for attending today's meeting. I know that we didn't have much time for the getting question for Muchun and Blessing, so we can continue it in the next month, or you can contact them directly. I will share the email details with them. And recordings I will upload tomorrow or today, I think it will be available for you. I will share with everyone. For the next month meeting, I will let you know that who will be presenting and when with the date, I will share with you everyone. And another quick note for the boot camp, I propose for everybody that we will give a 1 hour session for trainees to come up with the idea that they like to have. So I will share more detail coming weeks that you can have whole 1 hour with faculty members, whatever you like to discuss with them. So we will share more information about that. You can organize, and topics is based on you. We are not having any kind of disturbance to you. So it is purely your 1 hour. You can come up with the ideas during the boot camps. Think about that. I will share more detail later. And thank you very much for everybody. Today is, I think, the first day that we have the highest participation. Thanks again for that. Highly appreciate that. So see you in the next month, and looking forward to send an email with the time for the next month and also the upcoming detail about the boot camps. Thank you. Bye-bye.
Bye.